Between every two neurons you see here, there's a tiny gap. And it's this gap that allows the transmission of the impulse from one neuron to another. This gap is called a synapse. And that's the focus of today's discussion. A synapse is the junction between two cells and serves as the main point of communication between a presynaptic neuron, which sends the signal, and a postsynaptic cell, which receives it. And this connection doesn't only happen between neurons, it can also occur between a neuron and an effector cell, like a muscle cell or a gland cell. Synaptic transmission can occur in two main ways, chemical or electrical. And based on how they function, synapses are classified accordingly. Chemical synapses are the most common type in the human nervous system. Electrical synapses, on the other hand, are more prevalent in non-mammalian species. Chemical synapse, the more common type. A chemical synapse is made up of several key parts. Terminal bouton or axon terminal, located at the end of the presynaptic neuron. It's filled with synaptic vesicles and voltage-gated calcium channels. Neurotransmitters, chemical messengers like glutamate or GABA, synaptic cleft, a narrow space between the two cells, postsynaptic membrane, lined with receptor proteins. These can be ionotropic, ligand-gated ion channels, or metabotropic, G-protein-coupled receptors triggering internal signaling. So how does the signal actually cross? When an action potential reaches the axon terminal, calcium channels open. Calcium floods into the terminal, triggering vesicles to fuse with the membrane and release neurotransmitters, a process called exocytosis. The neurotransmitters then bind to receptors on the postsynaptic membrane, opening or closing ion channels. If the effect is excitatory, the inside of the postsynaptic cell becomes more positive. This is called an EPSP, excitatory postsynaptic potential. If it's inhibitory, the cell becomes more negative. That's an IPS, inhibitory postsynaptic potential. To prevent overstimulation, neurotransmitters are quickly cleared from the cleft. This happens by enzymatic breakdown, reuptake by the presynaptic neuron, or by diffusion, resetting the synapse for the next signal. Now let's talk electrical synapses. Unlike chemical synapses, electrical synapses allow direct electrical communication between neurons via gap junctions. Gap junctions are protein channels that link two adjacent cells, letting ions flow directly from one to the other, no neurotransmitters involved. At the core of each gap junction is a connexin, a channel made of 6 connexin proteins. These form a continuous bridge, allowing ions and small molecules like ATP or second messengers to pass through. Because there's no synaptic delay, signals travel incredibly fast. And unlike chemical synapses, electrical synapses can transmit in both directions. All right, let's break down the key differences between chemical and electrical synapses, side by side so it's easy to follow. 1. Transmission. Chemical uses neurotransmitters. Electrical, direct ion flow. Second speed. Chemical, slower. Electrical, much faster. 3. Direction. Chemical, unidirectional. Electrical, bidirectional. 4. Modulation. Chemical, highly adaptable. Electrical, minimal modulation. 5. Structure. Chemical has a synaptic cleft. Electrical, membranes are physically connected. 6. Plasticity. Chemical, highly plastic, supports learning and memory. Electrical, less flexible, but great for speed. 7. Where they're found. Chemical synapses are widespread, found throughout the brain, spinal cord, and peripheral nerves. Think neuromuscular junctions, cortical circuits, and limbic system pathways. Electrical synapses are found in specialized regions,